Hello, everyone. Welcome to the webinar. I'm Ann Thayer, contributing editor to the CNN Media Group, and I'll be moderating today's event. This webinar is entitled HILIC Analysis for Polar Modifications of Monoclonal Antibodies and is being sponsored by mAbs. CNN works with sponsors to identify topics of interest and value to CNN's audience and consistent with CNN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you're disconnected during the webcast, please log in again according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I'll be posing as many as time permits. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNN Online after the live webcast. The presentation today is being sponsored by mAbs. mAbs' mission statement is simply smarter chromatography, which means that when it recommends an HPLC or UHPLC product, it partners with customers to offer innovative solutions to complex problems. The company uses advanced analytical software to match its product portfolio with the customer's separation needs. It leverages 30-plus years of experience in the LC column business and its technical expertise in manufacturing network to help solve even the toughest application problems. And mAbs provides up-to-date and accurate technical catalogs and reports from leading separation scientists to keep clients informed of new technologies. During today's presentation, we'll hear from Dr. Barry Boyce, Chief Technology Officer at Advanced Materials Technologies in Wilmington, Delaware. Barry completed his PhD in neuroscience at UBC in Vancouver, British Columbia, Canada, then joined the DuPont Company in Wilmington, Delaware. Over the past 3 decades, Barry has enjoyed a variety of scientific, senior management, and executive roles with large corporate and smaller startup companies. In his current roles at AMT, the University of Georgia, and at GlycoScientific, Inc., his focus is on improving technologies for biomolecule separations, structural analysis, and functional roles. I'll now hand things over to Barry.
Good day, and thank you for that kind introduction. As mentioned, my name is Barry Boyce. Today I'll be discussing hydrophilic interaction liquid chromatography analysis, or HILIC, for polar modifications of monoclonal antibodies. Of course, everything that I'll talk about today with respect to polar modifications of monoclonal antibodies also applies to every protein that you're likely to encounter. But in the case of monoclonal antibodies, of course, there are good reasons why we have a lot of focus on post-translational and chemical modifications. Now, for an outline today, what we'll briefly touch on is some generic comments on monoclonal antibody structures, and this is, in today's discussion, really concerned with IgG antibodies, and we'll discuss certain types of polar post-translational modifications, particularly focusing on glycosylation, deamination, and oxidative changes to the side chains or modifications of the side chains of these proteins. We'll then conclude and have some discussion as you feel fit in terms of specific questions about the work that I'll be describing today. So as you can see in this slide, this is the common cartoon depiction of an immunoglobulin, in this case of isotype 1, so an IgG1 monoclonal antibody. Monoclonal antibodies of the IgG type are 150,000 molecular weight with 2 heavy chains, 2 light chains, connected at various points with disulfide bridges, both within the upper portion of the molecule in the so-called FAB region, between the heavy chain and light chain, as well as within each of those polypeptide chains, there are disulfide bridges. There's also disulfide bridges connecting the heavy chains in the lower portion of this molecule, the FC region, the constant region for each isotype of antibody. In terms of post-translational modifications, you can see in this cartoon, as well as the disulfide bridges, a depiction of the glycosylation site on the heavy chain at asparagine 297. There is considerable heterogeneity in the glycosylation of an IgG. This is depicted with cartoon representations of the oligosaccharides that reside on the asparagine side chain. So for today's discussion, we're really focusing in on the polar post-translational modifications. Unlike, for example, various lipidic or other non-polar modifications, there are a number of post-translational modifications, both chemical and enzymatic modifications of monoclonal antibodies that result in the addition or substitution of side chains with highly polar chemical moieties. And for today's discussion, this will include modifications at the predominantly at asparagine, so-called N-linked glycosylation, or through the alcohol-containing amino acyl side chains, O-linked glycosylation. These can result in the addition of a single saccharide or a oligomeric polysaccharide modification at specific sites within the molecule. In addition to discussing glycosylation, we'll also have some demonstration of the utility of HILIC for studying the deamidation processes, predominantly a chemical phenomena that occurs in all proteins, and coupled to deamidation at asparagine and glutamine residues is obligate isomerization of those now modified deamidated aspartic and glutamic acid side chains. As well, we'll briefly touch on a specific type of oxidation at methionine residues. And this is oxidation in the sense of specifically attacked by portions of dissolved oxygen. Okay, so the first concern will be for glycosylation. And when we think of protein glycosylation, and it's of course the same for all proteins that are glycosylated, but is particularly in focus for monoclonal antibodies. This refers to really any reaction that results in the addition of monosaccharides or polysaccharides to a protein. This is a biosynthetically coupled process, particularly for N-linked glycosylations. For O-linked glycosylations, this is a more dynamic phenomena with accessory enzymes that put on and take off O-linked modifications in a highly dynamic fashion. Why do we care about this for monoclonal antibodies? Because there is quite adequate evidence that this does impact the safety and in particular the immunogenicity of monoclonal antibodies that are going to be used for biotherapeutic purpose. And this is expressed through the efficacy of treatment using biotherapeutic monoclonal antibodies and including effects on clearance of those monoclonal antibodies once they are used in the therapeutic setting. That is elimination and metabolism of monoclonal antibodies. In terms of chemical processes, that is to say the storage and appropriate formulation of therapeutic molecules, there is a clear impact on stability and solubility in various formulations and under various storage conditions. So glycosylation is increasingly considered a critical quality attribute, and the glycosylation status of monoclonal antibody must be characterized as part of the development of that molecule for therapeutic purposes. And everything I just said about the use of monoclonal antibodies for therapeutic purposes is also true for diagnostic applications, but in a slightly different way. So the most popular application of HILIC for the analysis of glycosylation is an indirect approach whereby oligosaccharides are released from the intact protein using a particularly useful and robust enzyme, PNGase F. The released oligosaccharides or glycans, end-linked glycans, and I'll use those terms interchangeably, are then typically labeled at the chain termini through a reductive amination process. In my lab and many of my collaborators, we have found the use of procainamide to be a useful label. There are many other labels out there, and these are used in reductive amination processes, and there are other processes other than reductive amination that have been developed. This is a broadly popular technique, and there are very many methodological variations. In our case, we found that the use of procainamide as label and introducing it via reductive amination is useful and can be quantitative. The reductively aminated products can be well resolved using HILIC, and HILIC is exquisitively sensitive to the composition of the released end-linked glycans and is also sensitive to the branching sites within those end-linked glycans, so that we can get resolution not just of the different compositions of glycans, but also the branch variants for the complex structures of end-linked glycans. And this can be branches both within the core structure as well as in the sialated, the end antennary sialation sites and their branching variants. So there's been a lot of work done on this. It is a highly reproducible and predictable approach to the separation of these complex molecules. Now, in typical monoclonal antibody, you're going to get a mixture of end-linked glycans at that asparagine site at 297. Typically, you'll see the variants that are circled here, the G0, the G0F, the G1F, and G2F. You'll also see a number of sialated, less common variants or lower abundance variants at that site. So there's a lot of complexity in glycosylation that occurs in monoclonal antibodies. And HILIC has well lent itself to this analysis. Now, in this case, we're using mass spectrometry for the detection method. This gives us the ability to distinguish and confirm isomeric mixtures of the N-linked glycans. High sensitivity analysis can also be achieved through the use of fluorescence detection post-column as well. So this gives us an idea of the complexity and importance of using HILIC for N-linked glycosylation analysis. N-linked glycosylation is not the only modification that occurs within proteins. An important modification that occurs in other proteins, many of them important to human diseases, are O-linked glycosylations. Now, in the case of O-linked glycosylations, although it's possible to release the O-linked glycan, it's usually a single or a smaller oligosaccharide than typically is seen on N-linked modifications. Frequently, you'll do the analysis of the glycosylation, the O-linked glycosylation, while it is still attached to the peptide. Why do we care about O-linked glycosylations, and why do we care in particular about the so-called O-GLICNAC modification? Well, in the case of O-GLICNAC, there's been a lot of interesting biochemistry developed, and it teaches us a bit about biological control processes. O-GLICNACylation is the term that we use. It's a reversible modification at serine and threonine residues. In other words, the alcohol side chain aminoacyl residues. O-GLICNACylation is a modifier of biological activity. In other words, it affects, in particular, protein-protein interactions and activity of coupled enzyme systems that are under control, usually with association of protein-protein interactions. In addition, O-linked glycosylation has been shown in some circumstances to actually be linked to phosphorylation. And phosphorylation is a biological control process that we consider of great importance when we think about the druggability of therapeutic targets. So the interactions here between O-GLICNACylation and phosphorylation, and there's been a great deal of biochemistry work done on this, particularly led by Dr. Hart at Johns Hopkins. And what's been observed is that, first of all, this affects a lot of different proteins. And many of these are proteins that we think about when we think about oncology, about diabetes, and about neurodegenerative processes. And what we find is that there's this linkage of the phosphorylation cascade, the reversible addition through kinases and phosphorylases of phosphate residues at the same or at nearby serines or threonines where we see O-GLICNACylation. So this is a highly dynamic set of modifications coordinated by the activity of these four different enzymes, adding and removing O-GLICNAC and phosphate modifications. So we care about this when we would like to have an analytical approach to establish the presence or absence of this O-linked glycosylation within a protein. Our approach to this started off by taking a look at which would be a better chromatographic separative strategy using reverse phase or HILIC. And in most of these cases, using a combination of both UV detection as well as online mass spectrometry. So on the top panels of this slide, you'll see the UV trace. On the bottom, you'll see the mass spectral signal. And what we're doing is comparing the ability of reverse phase mode of separation to HILIC mode of separation to resolve two peptide, or a peptide pair. In this case, the peptide is derived from the amyloid precursor polypeptide. There's a glycosylated sequence at threonine number 4 within this sequence, and the unglycinatylated homolog referred to as PEP. So we have a GPEP and a PEP depicted on the chromatograms. As you can see in reverse phase, under similar gradient steepness conditions, so increasing acetonitrile at 1% per minute, there is resolution of the glycopeptide from the peptide sequence. But it's pretty tight. They're pretty close by. If we look at the use of HILIC conditions, again with a similar gradient steepness, we can see that there's a much greater resolution between those two sequences, the glycosylated and the non-glycosylated peptide sequence. With, as we would expect, the oglicnac modified sequence eluting later in HILIC, earlier in reverse phase. In other words, the more polar modification causes greater retention in HILIC mode of operation. And this leads to well-resolved peptide from glycopeptide. So that's the case for this particular sequence. How true is this generically? So we studied this with 12 different peptide sequences and 14 glycopeptides derived from those sequences, in other words, modified at specific serines or threonines with oglicnac or ogalnac. So what I'm saying is really focused on oglicnac because that's where we've been most interested in the biology, but it also applies to any OHEX-NAC-related modification. And what we see in reverse phase is that in many cases we can get successful resolution of the glycopeptide from the peptide sequence. And this is the delta RT for reverse phase towards the middle of the slide. The blue box, you can see the negative number means that the glycopeptide precedes elution of the peptide. Okay? In the case of HILIC, those are positive numbers, the delta RT HILIC, glycopeptide minus peptide. You get, first of all, larger retention shift, and this is reflected in the third column under RP results and HILIC results, where we're looking at resolution of the two peaks. So if we look across this entire range of peptide sequences and the modification of that sequence, we see that generally resolution is increased by threefold using HILIC compared to reverse phase. And there's lower variance in the relative retention in HILIC versus reverse phase. So the resolution is higher. It shows less variability based on the sequence. And in the red highlighted boxes at the bottom, you can see when we express this as a statistical measure, percent relative standard deviation, it's about a third as variable using HILIC relative to reverse phase. Now, what that means in words is that HILIC will do better at resolving these polar modifications to the peptide sequence. It's driven largely by a better separation selectivity difference when you put a glycan onto a peptide sequence. And again, this is true whether you're looking at oglicnac or any of the O-glycosylated sequences. Well, what about N-glycosylated sequences that are left on the polypeptide backbone? In other words, glycopeptides versus released glycans. Well, here we see what happens in a reverse phase experiment. In this case, a tryptic digest of ribonuclease. Ribonuclease contains a mixture of high mannose sequences at a small tryptic fragment that's generated from the intact protein. And in reverse phase, that small peptide sequence doesn't give you a lot of retention, but most importantly, it doesn't give you resolution of the glycan variants. In other words, when we look at the selected ions to distinguish the glycovariants, there is very little ability to resolve those glycovariants in reverse phase mode of operation. When you do the same experiment using HILIC, and I direct your attention toward the right side of this chromatogram, you can see color-coded for each of the glycovariants that they are now well retained and well resolved chromatographically. So this is the 5 different, the mannose 5, 6, 7, 8, 9 sequences that are found in ribonuclease B variants. Okay, so how true is this when we look at larger or more complex end-linked glycans? The top panel on the left-hand side shows a separation by HILIC of released N-glycans. So we've treated these with TNGase F, we've labeled them using procainamide, and then we've resolved the resulting glycans using HILIC. And what we see is the compositional as well as positional variants that you can obtain using HILIC with released glycan analysis. And these are different based on, the peaks are well resolved based on the differences of the branch points for the glycans that are present at that N-glycosylation site. So we see this for the biantenary, shown as the first green peaks, as well as the triantenary and complex multitenary glycans that you see in fetuin. If we take that same fetuin, but now not use release by TNGase F, but look at a tryptic fragment with the N-glycans left intact on the peptide sequence, we can also resolve those isomeric mixtures, those compositional and isomeric mixtures, leaving the peptide on. In other words, the selectivity difference in HILIC is driven by that most polar modification, in this case, the N-linked glycosylation variants. And we get a similar resolution with the peptide on to what we saw when we enzymatically released and labeled those glycovariants. Okay, this is the case in fetuin, which is a good model system for glycoscientists. It's also something that we see for a molecule of greater interest for those interested in IgGs. In this case, the experiment is operated using serum-derived IgGs produced by protein A isolation, then tryptically digested and resolved as a mixture using with detection by scheduled reaction monitoring, so we can pick out the specific peptide and glycan precursor and then fragment it and look at a selective signal. So this is a triple quad type experiment. And what we see by HILIC is some resolution acquired by differences in the IgG isotype sequences, the peptide sequences, single amino acid differences between the IgG subtypes, but significant resolution acquired by the associated N-linked glycosylation at that central asparagine residue for the glycovariants that are present of the IgG2-3, IgG4, and IgG1 subclasses. So again, we get resolution based on both glycosylation composition as well as resolution of the isomeric glycans at those positions based on branch variant selectivity within that glycovariant population. Another example as shown here, in this case with hemopexin. This is work not done by myself or collaborators. It was done independently by another group where their interest was in comparing various commercially available materials, including one that AMT makes, the HILIC column, to the commercially available BEH amide and Zinc HILIC materials. And again, it's an SRM or a triple quad type of experiment where the N-link glycosylation is left intact on the peptide sequence. So there's some chromatographic resolution acquired by the peptide separation and dominated by the selectivity differences due to the polar modification, in other words, the glycovariants. And what we see is a pretty informative, information-rich resolution based on both peptide sequence and those glycosylation variants at each of the modification sites. So these are different N-linked glycosylation sites that are being analyzed. So it is possible using HILIC under appropriate conditions to gain considerable information, not just on the glycovariants present, but what's their actual site of attachment. In this case, the comparison shows that the HILIC material gives a much greater degree of separation and simplifies the corresponding LCMS analysis. What's most striking is the considerable simplification of the chromatograms by the much greater retention of sialated species. So the acidic terminal sialation of N-linked glycans really drives retention on the HILIC material and well resolves those based not just on composition, in other words, the sialated residue, but also its branch variations. So it's the differences in the surface chemistry definitely have an impact on retention as well as resolution of these N-linked glycovariants. And this is just another example showing it's not just it's not limited to sialation, or should I say, positional variance. In this case, we see separation based on where a fucosylated or where the fucose residue resides. In other words, these are isomeric glycovariants. They have the same molecular weight, they're on the same peptide site, but they're different from where the fucose residue sits at its typical place at the near the asparagine site of attachment versus in the antenna on that biantennary species where that particular fucosylated residue sits in this glycovariant. Depending on the surface applied, in other words, the column material applied, you may or may not get full resolution of those. In the case of the pentahilic, they're well resolved. Partly in the BEH amide material, not well resolved in the HILIC. And there may be some thinking we can do in terms of, well, why would they resolve under particular condition or on particular surfaces and not under others? And this is most likely a reflection of the actual structure of the chromatographic surface and the local electrostatic interactions that occur on these neutral, in the case of the pentahilic and BEH amide, neutral bonded phase materials versus the HILIC, which has ionic modification. And we see another example basically showing the same, with the pentahilic showing a particularly beneficial ability to resolve these positional isomers. In this case, with the sialylated glycovariant. So the range of separation turns out to be better with the pentahilic material compared with an amide type of material or with the HILIC ionically modified type of HILIC stationary phase. Now, I'm going to leave glycosylation. We'll talk now about deamidation, another polar modification that occurs readily within proteins, and particularly is one that we see in monoclonal antibodies. So with deamidation and isomerization, what am I referring to? The reaction that we're concerned with is particularly observed at asparagine side chains, but also occurs at glutamine side chains. In other words, at amide side chain within proteins. The chemical process involved is a spontaneous liberation of ammonia from asparagine to form a cyclic succinyl intermediate. As I'll show you in a few minutes, this is readily visible with high-resolution mass spectrometry and LCMS analyses. This is, again, a spontaneous chemical reaction that occurs in all proteins, but is highly dependent on the sequence context as well as the pH that the protein's sitting in and the temperature either for storage or for incubation. This is a common reaction that occurs with tryptic digestion conditions. After the formation of a succinyl intermediate by this deamidation, this loss of ammonia, there's also the loss and addition of water that occurs spontaneously. If the water is added from one side of that succinyl peptide intermediate, you get back asparagine. So that would be relative to the, aspartate, relative to the asparagine, that would be a 1 mass unit difference, an increase in 1 mass unit. In equilibrium with that, succinyl peptide is also addition in the other direction of the succinyl peptide backbone bond. This gives you the isoaspartyl residue. Again, a +1 addition for mass. Now, the important difference between a aspartate and isoaspartate is that isoaspartate results in a main polypeptide backbone shift. In other words, the polypeptide backbone now has a methylene group in it that it didn't have before. And this has a direct effect on conformation. So it is important to follow deamidation and isomerization reactions in intact proteins, because it does have the potential to change the conformation of the protein directly and to change its immunogenicity and other processes. It's important to appreciate that this is a spontaneous and irreversible reaction. The first step is irreversible, the loss of ammonia, but everything after that is in equilibrium. In other words, you have a equilibrium between aspartate and isoaspartate. It's readily able under aqueous conditions to go back and forth between those structures. And as you can see, when you go from an aspartate, from asparagine to an aspartyl residue, you have a change in charge. You now have a carboxylic acid, which was previously a neutral functional group, but also you have a change in mass. So again, there's a potential here to reduce bioactivity and attack proteins. There's definitely an opportunity to change the pharmacokinetics, in other words, clearance and metabolism of a monoclonal antibody or of any therapeutic protein, and to change its antigenicity, particularly because you're now modifying the polypeptide backbone directly. Okay. Also, once you do introduce an isoaspartyl or an aspartate side chain, you definitely can get differences in terms of enzymatic sensitivity through normal degradative processes of the polypeptide. Okay, so we've studied this in both isolated peptides, model systems, as well as intact proteins. If you look at a typical humanized monoclonal antibody, there's a number of sites that are known to be present in the conserved regions. There's many that could be present in the variable regions, which is generally where you try to design changes in a monoclonal antibody. We've studied this in greatest detail with trastuzumab, looking at tryptic-derived fragments in the heavy chain as well as light chain sequences. And these are the peptide sequences that we've looked at. There's a couple of sites that we would expect to see some particular activity. And these are the ones that have a glycine residue C-terminal through either asparagine or aspartate. And these are highlighted here. Our model system is an IgG peptide that has multiple asparagines, 3 of them at 388, at 393, and at 394. The first one has a glycine residue C to the asparagine. The next two are neighboring asparagine residues, and we wanted to see what effect that would have on the rate of deamidation of those asparagine side chains. Okay. So this is the model system studied, again, comparing HILIC versus reverse phase. And what we wanted to find out was, is HILIC a better way to look at deamidation? You would think it might be, because this is a polar modification. And although reverse phase is perfectly capable of resolving polar modifications, it may not be as good as HILIC is. And what we find, using, in this case, 3 synthetic peptides that were synthesized from these IgG1 sequences, one with asparagine, one with aspartate, one with isoaspartate, is that, yes, you well resolve those by HILIC, you get less resolution by reverse phase. What we see in HILIC is we always get the same retention order, always asparagine followed by aspartate, followed by isoaspartate. And we see a similar situation if we deamidate glutamine, glutamine followed by glutamate, followed by isoglutamate. Okay, so HILIC has particularly good predictability in terms of the retention position of modifications due to deamidation. When we study the this model system, what we did is, of course, retention map them, define them in terms of their mass spectral properties, and this includes both MS and MS-MS mode, using both collisionally induced association as well as ETD. So we're able to distinguish exactly what the modifications are at the asparagine residues. And what we find is that after relatively modest incubation of the parental species, in other words, the asparagine-containing sequence, under mildly basic conditions at physiological temperature, you get rapid deamidation, and it doesn't limit itself to that first site at 388, that's next to the glycine. It continues and continues to the asparagines that are next to each other, further C-terminal word in that peptide. So we get a very complex mixture from this model peptide. And we assume this occurs in the intact protein. So we've done that experiment as well. Again, we are able to map these out. What we find is that at the end of it, what happens is you get an accumulation of a di-deaminated species. Predominantly, if you let it go long enough, you're going to get what's essentially a mixture of isoaspartate and isoaspartate formation as the dominant species. And we monitor this using high-resolution mass spectrometry, looking at the mass shift, as well as the MS-MS effects. Okay, so we did the same experiment with the intact protein. In this case, the stress was incubation of TH9 in the presence of mildly alkaline tris buffer. And then we studied this after tryptic digestion under conditions to minimize deamidation, and then looked at how much deamidation is going on and what are its features. Okay, so this is, for example, this particular peptide, the one that we've been using in our model system. What do we see in an intact monoclonal antibody? And we looked at 9 particular sites where we thought there might be some activity going on. Well, the first thing we saw was deamidation in the intact protein without asparagine-glycine pair. We see it in the native species, in other words, before we've incubated it at all under alkaline conditions or mildly alkaline conditions. When we stress the intact protein, do the tryptic digestion, and look at the results, we see considerably greater formation of the isoaspartate and aspartate residue. Also, on the bottom in the blue trace, you can see that cyclic intermediate supports that equilibrium reaction or interconversion between aspartate and isoaspartate. When we look at a different sequence, again, in this case, it's not an aspartate or an asparagine residue, so it's not a deamidation reaction. It's a spontaneous isomerization reaction with an aspartyl residue in the parental sequence next to a glycine. In this case, what we see is that in the intact therapeutic preparation, there is already isoaspartate present. And we see the cyclic intermediate present. After stressing, in this particular case, there's no change in the amount of isoaspartyl formation. Okay? In a way that makes sense. It gets to its equilibrium distribution and then stops. And again, diagnostic of what's going on is the presence of that cyclic intermediate. Okay, I'm just going to very briefly, because the hour is getting later, very briefly touch on oxidation reactions. In this case, the one I'm focusing on is oxidation of methionine. So methionine with that sulfur group in there is subject to oxidation there's actually two different levels you can get oxidation with a singular addition of an oxygen at the sulfur atom, the methionine sulfoxide. Or you can get two oxygens added to form the sulfone. In the case of oxidation of monoclonal antibodies, we know from existing literature and history that methionine oxidation in monoclonal antibodies is definitely linked to functional loss of activity, bioactivity, that there are quaternary and tertiary structure changes, and that this drives a propensity to form aggregates of the monoclonal antibody. So, direct effect on production of the desired monoclonal antibody, how to formulate it, and how to store it. Okay, in this case, we've used various model peptides. This one from BSA, by reverse phase, not terribly well resolved. When we look at HILIC, much better ability to resolve those polar modifications by LCMS. And what we see predominantly is the sulfoxide. Very little, if any, of the sulfone. In this case, the example is from mAbs. We're looking at the formation of the methionine sulfoxide, clearly present as a delta 8 in the mass spectrum because it's a +2 peptide. Well resolved using HILIC conditions. So, to conclude, we're clearly getting better at characterizing post-translational modifications, particularly in therapeutic monoclonal antibodies, where there's been a lot of attention. HILIC separations show particular facility to improve the resolution of polar post-translational modifications versus the unmodified materials. And this is for glycosylation reactions, for oxidations, for spontaneous deamidation. So it is showing superior resolution compared to reverse phase. We have a good way to do this. The methods have been found to be robust in terms of separation and can be quantitative. And with that, I'll acknowledge the various collaborators on these projects, Andrew Heron, Ariane Solvian, Ben Libert, Connor McHale, and Stephanie Rosenberg, Stephanie Schuster at Advanced Materials Technology, as well as my longtime collaborator at the University of Georgia, Ron Orlando, and his graduate students. And with that, we'll move over to some questions. Thank you.
Great. Thank you very much, Barry, for that very interesting presentation. Quickly, before we do some Q&A, we'll do a quick poll question for the audience. So the question is, would you be interested in learning more about the Halo BioClass columns? Just please mark your answer on the screen and hit submit, and we'll just take a few seconds to answer this one. Okay, if you'd mark your answer, that's great. We'll move on to the next poll question, which is, do you feel like you learned something new about biopharmaceutical separations or method development today? Please just answer yes or no and hit submit. Okay, now we'll move on to some Q&A for Barry. First question is, someone asks, how did you analyze the native protein?
When you say native protein, you mean how do we analyze the native proteins, or how did we analyze the? Okay, so I'm going to answer that two ways. We do analyze native proteins, but less so by HILIC. For many intact proteins, including monoclonal antibodies, HILIC methodologies have not yet been well developed, to my knowledge. We predominantly have used reverse phase methods for the analysis of fully intact proteins, but that's a slightly different discussion than we've undertaken today. In terms of the experiments that we've done looking at the amidation on intact proteins, we do have a tryptic digest analysis that is in that workflow. So, for example, when we look at the effect of stress on deamidation, in other words, we're stressing the intact monoclonal antibody, we are then tryptically digesting it and looking at the result from that. In other words, at the glycopeptide or glycan level. I'm sorry, at the peptide level.
Great, thank you for that answer, Barry. Someone else asks, have you ever evaluated column lot-to-lot reproducibility for the same analyte for HILIC versus RP?
Yes, we have, and in fact, AMT makes a product referred to as the HILIC glycan column. Each of those columns, each of the lots of material are qualified using N-linked released glycans so that we can control the variability for glycan analysis from lot to lot of material. Typically, under HILIC separation conditions for released glycans, there is less than a tenth of a minute retention shift between lots of material. That's for actual labeled released glycans. For glycopeptides, we do not specifically specify the product for that in terms of actual metrics, but we found very little difference between lots of material when we've applied glycopeptide analysis.
Great. Thank you. Another question is, what other amino acids, in addition to methionine, do you think undergo easy air oxidation in proteins?
Well, you can get oxidation at the cysteine residue. There's no question about it. Usually, when we think of oxidation reactions at cysteine, we think about reduction of disulfide bonds. But if you have molecular oxygen present, you can definitely oxidize the sulfhydryl directly. But there's many other oxidation reactions that can occur in proteins, including at aromatic residues in addition. So, those are less typically observed in biotherapeutic preparations, but they're not absent. But it takes more aggressive conditions to demonstrate them. I mean, typically, the workflows that show those oxidation products will also include the addition of peroxides and/or metal cofactors that will catalyze the oxidation reactions. Actually, I think you can even do that at asparagine if I think about it carefully. So you can, if you put enough oxidative condition in, there's many amino acyl side chains that can be oxidized.
Okay, thank you for answering that. Someone asks, are HILIC gradients typically opposite that of RP?
Well, it started off in history of HILIC that was an easy way to explain it. That, it uses the same mobile phases, but you just run the gradient in the opposite direction. That's obviously an oversimplification. And the features of proteins or protein fragments that drive retention and the subtlety of retention are a little different in reverse phase than they are in HILIC. So when we think of reverse phase, for example, there's very little sensitivity to glycan site occupancy in terms of retention processes. A small glycan versus a huge glycan can show almost the same retention in reverse phase. But under HILIC conditions, huge difference in retention, huge difference in selectivity. So we, for simplicity's sake, in a lot of lab work that we do, in my lab, what we do is we use a formic acid ammonium formate mixture as the aqueous, and a formic acid modified acetonitrile as the strong organic. Now, in reverse phase, we use the term strong organic. In HILIC, it is, of course, the weaker of the mobile phases. So the start points and stop points are usually not identical in HILIC versus reverse phase, but the gradient conditions are usually somewhere between 0.5% per minute acetonitrile and 1% or 1.5% per minute of acetonitrile. They're not perfectly inversion one to the other. Hopefully that helps. Hi, anyone still there?
Oh, sorry, Barry. We're just going to take one more question because we're running out of time. When you use a HILIC column, do you think ammonium formate is better than ammonium acetate?
I generally, for peptides, prefer the lower pH conferred by ammonium formate. But in terms of mass spectral sensitivity, there's very little difference in my experience. And they're both available with very high purity in LCMS forms. For solubility purposes, particularly for tryptic digests, we tend to prefer the lower pH that is supported by formate buffers or ammonium formate formic acid mixtures instead of acetic or ammonium acetate mixtures.
Great. Well, we're going to wrap things up there because I'm afraid that's all the time we have. Thank you again, Barry, for your very interesting presentation. Thanks to all the participants for being a great audience. Please be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars and the replay of this webinar. Our thanks to On24 for technology and production services, and thank you to mAbs for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Ann Thayer. Have a good day.